for you to confirm your name and your title. We'll start with that.
So my name is Dr de Koning and I am professor in animal breeding at the Swedish University of Agricultural Sciences. And I'm based in Uppsala in Sweden. Because my first name, Dirk Jan, is not always that easy to pronounce, I have been known as DJ in my professional role for several decades now. So that's often what I prefer to choose as my given name.
Thank you for that. Okay, so can you tell us where you are right now? Where are you sitting, DJ? What can you see?
So I'm in my office. I'm actually in one of my two offices. I'm in my office at the faculty building. So I'm now today in my office in my role as Deputy Dean of the Faculty of Veterinary Medicine and Animal Sciences at our university. And when I'm looking out of my window, I see our big lecture building. That is a building with our biggest lecture hall with up to 500 people and lots of smaller lecture halls. And when I stand up and look a little bit more, I see lots of students moving about because that has really started just this week. The students are back on campus for real now. So it's a bus with students finding lunch everywhere. And then I see also the solar panels on top of all the buildings around me. So.
Well, that's good. It's quite environmentally friendly, and obviously all the students and the lecturers are coming back for the beginning of the autumn term. And you mentioned that you had another office. Is that in the same building, or is it just next door?
The other office is in the building next door. We have the faculty has its own research building where nearly all the research departments are based. There are two others in other locations in Sweden. And that building is also connected to our animal hospital, which of course is very important for our veterinary medicine program and research. We also have a research farm outside the city and research farms in other parts of Sweden. But my research office, so to say, is in the building opposite this one with, where most of the colleagues are based.
Right, so you've got a bit of a choice of which one to use, and you don't have far to commute between the two.
Indeed.
That's good to know. That's good to know. So let's talk about Work Package 10, part of the Defend project. And basically, if you can tell me what the purpose of Work Package 10 is. Obviously, we know the title of it, but just in terms of what it aims to do, what it hopes to achieve, and how it started and how the work's going.
Yep, definitely. So, of course, Work Package 10 focuses on lumpy skin disease. This is a disease that we think is spread by stinging insects, and it's gotten its name lumpy skin from the nodules that appear on the sick animals. Now, like with many other diseases, we see that when we have an outbreak, some animals are severely affected and others are not. And this is what we see for a range of diseases across species, including, for example, COVID in humans. We also see variation there. And from my research, the main interest is really find out, okay, these difference that we see between individuals, is that due to genetics? And how much of it is due to genetics? And if there is a proportion of this variation that we see due to genetics, is that based on a few genes with a large effect, or lots of genes with tiny effects that add up to bigger effects? And that is really where we got into when we were invited to take part in Defend. This observation that we already had, the colleagues had from field studies, that about 50% of the animals get ill in an outbreak, but that could also be due to the fact that not all the animals get exposed to the virus to the same degree, but actually the same was observed when people did controlled infections in the laboratory, like in Pirbright or Cienzano and other institutes, that even when all the animals are infected using the same protocol, only about 50% was infected. So then that gives a good idea that maybe there is something genetic behind this variation. And so my curiosity really, of course, we want to know that, but it's not just interesting science. It's also, if we know there is a genetic component, we can use selective breeding to breed the next generation of animals that would suffer less from disease outbreaks, such as lumpy skin disease. We have very good examples that, like the bovine TB that you still have in the UK, there is a very extensive selective breeding program underway to reduce the incidence of bovine tuberculosis. So, and that is really the whole stretch. And this is quite a big ask of what we are trying to achieve in Defend. We start asking the question, is there genetic variation behind this variation we see? Can we identify it? And on the basis of, can we find underlying genes? Is it 1? Is it 100? Is it 1,000? On the basis of what we find, how could we use that in selective breeding? So ideally, by the end of Defend, we would be able to say that, okay, genetic variation explains this proportion of the variation in resistance or susceptibility to disease. So like from the variation we observe 50 or 30 or whatever percent is due to genetics. And then we have identified, hopefully, that it is few or many genes. And on the basis of that, we can make a recommendation on the basis of extensive computer modeling on how can we use selective breeding as one of the many tools to reduce the impact of lumpy skin disease in the future.
I mean, it's so important, isn't it? It's got the potential to be quite influential on the farming industry, obviously breeding. With the scale of what it is that Work Package 10 is sort of hoping to achieve, as you say, to get to this endpoint and to find an explanation of why this sort of, I guess it's a kind of immunity, isn't it? Sort of, being able to see that about 50% are affected, and roughly 50% remain sort of unaffected by lumpy skin disease, even when they're directly exposed to it. So if we get to that explanation, and what I'm trying to say is that obviously you've got such a great piece of work on your hands here. It's a very large part of the Defend project, and I wonder, what is your approach to this? How do you, when it's difficult, how do you stay motivated? What it is that gets you going in the morning? How do you approach your work?
Okay, I'll first talk specifically about how we approach this in Defend, and how we also have changed tech. Because when we started Defend, when we started thinking and writing Defend, there was still outbreaks within the European Union, within the member countries that came into the project. And the first idea that we had in Work Package 10 was to have a large population-based study that we would collect lots of samples from within the project, from outbreaks that had happened, from both animals that suffered from disease and animals that did not appear to get ill, and build up a large case control study and focus all our research efforts on that. And now, for a variety of reasons, over the first 2 years of Defend, it became apparent that we were going to struggle to get that large number we had in mind, partly because within the European country, the vaccines were doing a good job, so the outbreaks were not happening, partly because there wasn't as much biobank material as we'd hope there would be. Thirdly, because the countries where the outbreaks were happening, they either had difficulties either collecting samples or getting permission to send them to us, because there's politics as well. And finally, also one of the large experiments that was planned where we would get about 250 samples did not happen in the end. So already in the earlier stage, we're thinking, okay, if this study does not get to be as big as we'd want it to be, what else could we do? And then so we added a big dimension to work package 10, and that was to move to, beside the population study, which we still did, albeit on a smaller scale with some promising results. We also moved to look not just at genetic variation, but also at how the genes that are involved in these processes are expressed in individuals that get disease or individuals that stay healthy. And for that, we were very fortunate. We have both Pirbright and Shenzano in the project, where studies that were already planned and performed, like artificial infection studies with lumpy skin disease, also have very good practice to biobank samples. So both from experiments that were done within Defend, plus other experiments that had already been done, we could, on the basis of biobank materials, plan to look at the gene expression in different tissues, in different animals, potentially, depending on whether they got sick or not. So that was one of the big changes our work package made halfway or even before that. And that also meant that, and that has been, so we have one experiment there in Belgium that we have completed. We will do another one there with data from Pirbright that is being generated as we speak. But it also meant that we could actually broaden the scope of work package 10, because then we realized we can do this for LSD, but we can even do this for African swine fever, where in work package 7 we have ample experience, and we also there have past experiments that have biobanked samples. And this is, I think, a very important part of what we do in work package 10. We do not require additional animal experiments, but we add value to data that is already being collected, and we make more use of samples that have been biobanked as part of good practice animal experiments. So there's a very clear 3R argument there as well. So what approach?
I was going to say the biobank, aspect of this is obviously really important, and it saves conducting, new experiments and sampling from newly gathered animals. But I was wondering about this. So when you look at the samples, do you need some of the biobanked material to come from a particular obviously it's cows, but within like a particular breed of cow? Or can you look at, like when you have this biobanked material and you might have, I'm not sure exactly how this works, but if you had, for example, material from a farm in, say, Scotland, and then you're comparing it to a breed of cow in Italy or somewhere, does that have any influence in terms of the genetics, because they've obviously had sort of different come from different areas, come from different genealogical lines?
Yep. So that's when we do the population study, or what is called this genome-wide association study that we have completed recently. We have material from a lot of countries and a lot of different breeds. but within the country and within the breed, we should have both cases and controls. So as long as it's sort of, we have that balance that we have within the breed, we have both the animals that succumb to the disease and those that stayed healthy, then we're okay, because all these genetic differences because of population differences, that's the first step we do when we have all the genetic data. We look like, okay, how is this, what is the genetic structure of this data, and how does it relate to country of origin or breed of the animal? And then we see very clear clustering. And then when we subsequently go along the genome to find individual genetic markers that may be correlated with resistance to disease or susceptibility to disease, then we simultaneously in that analysis, we model this underlying population structure.
It's so interesting, isn't it? I mean, do you have any idea at this time of how that where the results might lead you, in terms of, have you got any evidence at this time to say that there's particular breeds that are showing resistance or are showing susceptibility? Is it too early to say?
So we have looked because of the samples we had, we can only do an analysis across all the breeds if we put all the animals together. What I will say is what I find very motivating is that this sort of 50-50 split that we see between animals staying healthy or animals getting diseased, it's pretty much identical from when we look at controlled experiments in purebred on Holstein cows and when we look at all the country data that we have collected. So there doesn't seem to be much differences in incidence, and even some published studies that looked at sort of native breeds don't see quite the same. But and one reason for that might be that lumpy skin disease is new for all these populations. So they have not, it's a new disease for the Balkan countries. Of course, for the experimental infections, it's not in a breed where it's been before. So there has been no selection pressure for lumpy skin disease in our populations. Maybe if we looked in African cat populations more, where the disease has been endemic for much longer, maybe there was already a clear differentiation that overall they would be more resistant. But the fact that we see these similar patterns, to me, confirms that, yeah, these breeds haven't been exposed to this disease before, which also means that potentially the effects that may be hiding or that may be in the population related to susceptibility or resistance to this disease could potentially be quite large.
It's really interesting, isn't it? And let's return to this question, obviously, with the adaptability that was required from perhaps the original plan to sort of how you obviously had to shift to using more biobank material. That shows a certain dynamic approach, doesn't it, to work package 10, and being able to be flexible and like you say, change tactics. So what is your personal approach to the work, would you say?
Yeah, I'm an animal breeder or a quantitative geneticist, and my work, regardless of the species I'm working on or the traits that I'm looking at, I have always been this combination of traditional animal breeding plus using information on the DNA like we do in these genome-wide association studies, but also using information from the gene expression. So in that sense, what we did now with work package 10, of course, fits with an approach that I have been developing and using for some time now, where, okay, the DNA information can tell us a lot, but seldomly everything. So the DNA information can pinpoint some interesting areas, but then we need to add in functional information to really understand what is happening at the molecular level, if you wish. So that approach I'm quite familiar with, but here we had not planned for that in the beginning. But now that it turned out, based on the experiments that were done anyway, that we could add it in, it was an extremely good alternative.
It sounds like it's worked out really well. So can you tell us where you are currently?
We have now, towards the end of the project, we are getting a lot of data to work with, which is always the case when you're working on data analysis in the EU project. So we have completed the analysis of the experiments at Cienzano in Belgium, where we looked at gene expression over time for animals that stayed healthy and those that got sick after artificial infection. And there we found a moderate number of genes with quite strong effects. So there we have some leads to follow up, because when we look at the biology behind those genes, those that understand the disease much better than me see that, okay, yeah, it makes sense that you find these genes. So that's promising. And also, when we have completed the population study, the GWAS study, on the larger samples from the member countries, and there we also see that well, we don't have very much statistical power there, but we anyway get strong signals, which is encouraging. And that would also suggest that there is at least some larger effect. So both the study on the gene expression from Cienzano and the genome-wide association study suggest that there is a modest number of genes with a considerable effect. We're currently data is being generated from a larger experiment, or with more samples from Pirbright, and we're hoping to validate some of these findings there. In the meantime, having those preliminary results where we are now, we are building a computer model of what might be the genetic background of resistance to lumpy skin disease, and then how could we tackle this with different breeding strategies. So we have we're constant, and those are pretty much the same people in our group doing the analysis and doing the modeling. So they talk a lot to each other to sort of get to this big part. Okay, with this knowledge, what we know now, what recommendation would we make to breed for better LSD resistance in the future? So that is where we are now, and we're drinking from the fire hose when it comes to data, and then luckily we have sufficient time to synthesize that. But I'm confident that some of the fine tasting analysis on the utility of these results will continue post defend as well.
It's a great, it's a huge logistical exercise as well to gather all of that data to be able to build it into a computer model. That's really exciting. You mentioned work package 7 with African swine fever, and I wondered if you could tell us a little bit about how work package 10 fits into the wider context of Defend, and what other work packages it kind of connects with.
Sure. So one work package to start off with to name is work package 2, even though there's no real link in terms of data. The work package 2 is very relevant because it looks at the variation in the pathogens, and work package 10, on the other hand, looks at variation in the host. So they're very much the yin and yang there of host pathogen interaction. With work package 4 and 5, where the experiments that have been done primarily at Cienzano, they are very important for us to get an idea on, okay, because they give us more detailed information about disease progression and when can you see that the animal is going to be sick or going to stay healthy. And they are as sophisticated. They say, okay, after 3 days, we can already tell pretty precisely which animals from our experiment are going to be the ones that get sick. So that's so they have been very good for and of course Pip in that as well from Purbright, but they have been very good in helping our understanding, helping our planning, and providing the samples for some of the studies. So so 4 and 5 are big ones for LSD. For work package 7, of course, that's where the experimental work of on African swine fever, the experiments on from Purbright that we can bring into work package 10 when we look at gene expression. That's a strong link there. Work package 9 was initially planned to also include a large experiment in South Africa that would have contributed another 250 animals and sampling on farms, and that we had a lot of hopes to include a lot of data from there as well, but that fell through for a variety of reasons. And we're happy that we have the resilience in the consortium that we can do our project regardless there. So in that sense, we over the years have been in contact with many of these work packages.
You make a really good point there about having the resilience within the consortium, within our partners. And I was thinking again about how much of a collaboration Defend is, you know, across work packages with all the expertise that we have in the consortium, and obviously it's you know in many different countries, so it's largely global, and that presents all kinds of difficulties that you've had to overcome, I'm sure, and it's just a very it's very reassuring and very exciting that, you know, we have so much within Defend, and we have so much expertise that we can really find out so much about ASF and LSD. So to conclude, I really wanted to find out a bit about how you got into this line of work, because I always think it's quite interesting to find out from scientists, you know, why they chose to take a certain path, or what was it that interested you to this particular line of work, because it's obviously so specialized. And you mentioned your background, but I mean, how did you end up here? How did you end up sitting in that chair talking to me today?
That's a very, very good point. So when I was studying, for my degree in animal science and in animal breeding, I was for a long time convinced that I wanted to do the biotechnology. I wanted to work in the lab and maybe even with genetic modification. And then in 94, I had my first practical time within my studies. So I was 6 months in Finland, the research institute in Jokioin. And there I was working in the lab, but I was also working a bit on analysis. So doing a bit of both data analysis, but also hands-on lab work. Following that, I did an MSc project in Wageningen, focusing entirely on the lab work, developing microsatellite markers, which were at the time sort of the favorite marker of choice. So I was developing microsatellites in chickens. But then I got the opportunity, because I got sort of interested in the analyzing side more and more, analysis of things. So then I got to spend 6 months for a second master's project at the Roslin Institute in the beginning of '96. And after that, I was pretty certain that it was going to be the analysis side for me and not the lab side itself. I wouldn't say I was hopeless in the lab, but you'll have to take other people's references for that. So anyway, but from that date, already from my time in '94, when I was in Jokioinen in Finland, I was looking at the links between DNA variation and traits. So that was then called QTL mapping, quantitative trait locus mapping. I continued that theme in my PhD in Wageningen, where we were looking in crosses between commercial and exotic pigs and finding genome regions that explain these strong differences we see. Then I moved to Roslin to do more of the same, but then other species. And I've honestly really continued on that track, working a bit on methodology, working a lot with actual data, but also working on a broad range of species. So I'm not a species or trait specialist. I always work with colleagues who know far more about the species or the trait than I do myself, like we have in Defend. But I sort of come with my box of tricks, which is the genome analysis and the gene expression studies, and I sort of see how that can fit together. And when I've so now I've got projects in aquaculture, I've got projects in pigs, cattle, lots of work in chicken, laying hens, but it's sort of the approach to things rather than the specific trait or the specific animal, which also made it possible for me to come into a project like Defend and meet a whole host of people with new skills and another network. And as long as we can sort of talk each other's language, then I mean that technically, then we can make progress. But so I have moved around. I'm now in Sweden since 2010, but the area has very much been this livestock genomics sort of broadly.
I think I mean, it's sort of pointing out the obvious, but researchers and scientists obviously have a sort of curiosity, a natural curiosity to find answers. And I think, it always lies in the data. And there's two sides, because you have to get the data to start with, and then you have to analyze the data. But I think of Corolla. I remember in our podcast, we were talking about the disease detectives, and I think of you as the data guy. You're the data guy who wants to do the analysis and find out what it reveals. And that's why it's so symbiotic. All of our work packages, they all fit together in a sort of interlocking system, but they do exist in their own right as well. And that's what makes up the Defend project. So thank you for your time today.
Thank you very much. It was a joy to talk about the work.